Biological Hazards and Controls, presented by David L.A. Rawson, International HACCP Alliance, Chapter 3, 123foodhandler.com. So when we talk about biological hazards, it's important that I clear up a few things here. We have a couple things to look at: pathogens and spoilage organisms. So biological hazards are pathogens. What is pathogen anyway? Well, let's take a look at the definition of pathogen. Patho is the Greek word for suffering, and genes is the Greek word for maker of. Pathogens are the maker of suffering. Pathogens are illness-causing microorganisms. Spoilage organisms, like yeast, mold, fungi, and some bacteria, are not biological hazards. They definitely may affect the quality, but not safety. Rarely cause illness. However, there are a few exceptions, a few commodities of food that we need to know about, the spoilage organisms that do produce byproducts like toxins. Foodborne illness in the U.S. On an annual basis, there's 48 million cases. About 128,000 of those 48 million end up in the hospital, with approximately 3,000 people die each year. This information is based on CDC Outbreak Database and a study done by Scallon et al. In 2011. So let's take a look how they get that information. Well, a person becomes exposed. The person then, after 2-3 days, maybe 2-3 minutes, maybe a week, maybe up to 59 days from hepatitis A. Person seeks care. Specimen is obtained by the physician. The lab test for specific organisms. Case is confirmed if an organism is found. And it's then reported to the health department, which then in turn reports to the CDC. So we suspect the data we just looked at on the other slide, 48 million a year, 128,000 in the hospital, is probably a little bit off. So how do people get sick from food? Well, the foods that are commonly involved in outbreaks are most frequently foods of animal origin: meat, poultry, seafood. But more and more lately, increasingly, it's become contaminated fruits and vegetables. You see, when diets change, so do foodborne illness outbreaks. So how does a person get sick? Let's take a look at the steps necessary to cause an illness. Pathogen or a toxin is present in the food. In some cases, pathogen must grow in the food to a high enough number to produce an infection or toxin. Some pathogens, like E. coli 0157H7, have a very low infectious dose. Parasites and viruses do not grow in foods, they just need a ride. Food must be ingested and survive the digestive tract barriers, and some pathogens must establish and multiply in the body. Places where people get sick or acquire illnesses is most commonly a food service establishment and group feeding situations, banquets, buffets, things of that nature. Retail. Etiologic agents of foodborne disease. This kind of supports that study I talked about earlier. What are the causes or the agents that cause these diseases? Here's a table from the CDC confirming the top 5 illness-causing pathogens. Remember, a pathogen is a gene, a gene that causes suffering, right? Illness-causing organism. So we got norovirus, number 1 by far, 58%. Salmonella, non-typhoidal. There is typhoidal and non-typhoidal, or Salmonella typhi and non-typhi. You can see there it's number 2 on the list. And then we go on to Clostridium perfringens, Number 3 on the list. Campylobacter jejuni or Campylobacter species, 4 on the list, and Staphylococcus aureus, number 5 on the list. So these are the top 5 illness-causing organisms. So here's a pretty good list of the most common bacterial foodborne pathogens. Salmonella. It's like 2,600 species of that. Clostridium botulinum, Staphylococcus aureus, Yersinia, Listeria, Vibrio species, Shiga toxin producing E. coli, Clostridium perfringens, and Bacillus cereus. Campi, finish it up with Shigella, which causes the illness shigellosis, which I usually say is the grossest. There's really only one way to get shigellosis. You ate human fecal material. You ate a turd. There's some pathogenic viruses we need to worry about as well. Hepatitis A and E, and remember the number 1 cause of foodborne illness in America, norovirus. Let's move into the pathogenic parasites. Some of the parasitic worms like Trichinella and Anisakis, those are visible with the human eye. And then we move into some microscopic creatures, protozoa like Cyclospora, Cryptosporidium, Toxoplasma gondii, and Giardia. These are some of the common pathogenic parasites that impact food. And here's one you need to know about when you're in the beef business. Prions, particularly slaughterhouses. Smaller than a virus. Prions are thought to be the cause of a group of diseases called transmissible spongiform encephalopathies, TSE, which are diseases of the brain. Mad cow disease or bovine spongiform encephalopathy, BSE, is the brain disease that affects cattle, caused by prions, we think. So don't forget, this is all about preventing foodborne illness. And how do people get sick from food? Well, they either get an intoxication or an infection. Let's take a look at those a little closer. So with an infection, we ingest live organisms. Viable pathogens infect and multiply in the body. Examples, Salmonella, Listeria, and in some cases, these viable pathogens may produce toxins in the body, which is pretty scary. So what happens? Well, we eat something that has a lot of bacteria on it. The bacteria evades our defense mechanisms, like in our mouth, in our saliva, we have some antimicrobial material. In our stomach, we have a hydrochloric-like material, hydrochloric acid-like material, excuse me. So we hit them in the mouth, we hit them in the stomach, and a low dose of bacteria will be destroyed. But a large dose of bacteria, a high count of bacteria, live organisms, could cause an infection. It evades our defense mechanisms and causes illness. So another way we get sick is by intoxication, where we ingest toxins. At a certain level, these toxins cause illness, perhaps even death. In the case of Clostridium botulinum, high likelihood that you could die, one of the deadliest known toxins to man. So what happens is that the bacteria produces the toxin as a byproduct. And then there are some other toxins that are found in food, either naturally occurring by decomposition or naturally occurring as part of the animal. Toxins. We eat too much toxins, we get an intoxication. Could die. So we've learned so far that there's these microscopic organisms, there's some parasitic worms we can see, There's small things like prions that cause these disease and illness to humans. So we know what the causing agent is, but how does that agent get to us? How does it get to the food? How does it make us sick? Well, here's some of the factors that contribute to these diseases, to these illnesses. In the case of norovirus, the number one cause of foodborne illness, that's going to be primarily poor personal hygiene that causes that. We move to number two, salmonella, number two cause of foodborne illness. That's going to be probably more than likely cross-contamination or inadequate cooking. You move down into Clostridium perfringens, number three. Now we're moving into inadequate cooking, holding temperatures, improper cooling. So we kind of know what's going on. We've learned quite a bit about these things. So you can see here this list of factors that contribute to foodborne illness, foodborne disease, and the outbreaks. Bacterial forms, spore formers and non-spore formers. Spore-forming bacteria cause big problems for the food industry. Three species of spore formers, C. botulinum, Bacillus cereus, and Clostridium perfringens, are infamous for producing toxins. Spores are biochemically, structurally, and physiologically different from non-spore formers or vegetative cells like Salmonella, Listeria, and E. coli. So what's all this mean for you? Well, we need you or someone like you, someone knowledgeable, someone with understanding about food, meat and poultry, and the processing of food or meat and poultry, so that they can determine the pertinent organism. Is it a spore-forming organism? Is it a non-spore-forming organism? What contributes to this pertinent organism? Is there any association of the pathogen with a particular product? Have there been any outbreaks associated with this product? Is there any outbreaks associated with the ingredients? So there's an element of research involved here. So yeah, you got to consider the sources. Where are these biological hazards coming from? Well, based on historical data and information we know, beef, very common, salmonella, E. coli, and Clostridium perfringens could be involved or part of an outbreak. When it comes to poultry, it's salmonella, campi, and C. perf. And with eggs, it's salmonella. These are the pertinent organisms for those particular foods. So once we do our job identifying and evaluating hazards, then we determine that maybe some are significant or enhanced or introduced, we have to start looking at control measures. Here's some control measures for biological hazards. Well, some of the strategies. We want to prevent them. We want to eliminate them. We want to reduce them to acceptable levels. So we can prevent contamination of foods. We can inactivate the foodborne disease agent, or we can prevent the multiplication of pathogens. So here's another reminder of the factors contributing to foodborne disease outbreaks: improper storage and holding, inadequate cooking, poor personal hygiene, cross-contamination, improper reheating, and poor storage practices will enable the microorganisms to grow, to enhance, to increase the likelihood of causing an infection, to increase the likelihood of toxin formation. We want to make sure we're producing safe foods, preventing infections, preventing intoxications. So let's take a little closer look at some of these organisms. Let's start with Clostridium botulinum. It's a spore-forming organism. It's toxigenic, which means it's the toxins which make you sick. It's found in the soil and marine sediment, which means it easily makes its way onto vegetables and seafood. It's anaerobic, which means it grows and an environment with no oxygen. There is no growth below a pH of 4.6, and the spores are extremely heat resistant. So, how do we control Clostridium botulinum? Well, there's a thing called retorting, which is a very high temperature process, which works with pressure to destroy the spores, as well as we can use growth inhibitors or pH, lower the pH below 4.6, like we learned earlier, and water activity, maybe using salt or sugar or other ingredients, and temperature, keeping it frozen. So let's keep moving through. Clostridium perfringens, it's another spore-forming organism. It also is a toxin-forming bacteria. It's found in the soil, intestinal tract of animals, meat, vegetables, spices. It's another anaerobe, grows in the absence of oxygen. The optimal temperature for growth is 43 to 45 degrees Celsius, which is 109, 113 Fahrenheit. Very rapid growth in this temperature range, sometimes called the temperature danger zone. Heard recently can double as quick as every 8 minutes in this range. Well, when we talk about the control of Clostridium perfringens, we know the vegetative cells are easily destroyed at normal cooking temperatures, but the spores can survive cooking processes and then germinate later when it's in the temperature danger zone. So improper holding or cooling could allow growth. So we want to make sure we hold and cool properly to prevent the growth of Clostridium perfringens. Remember, Salmonella, the number 2 illness-causing microorganism, happens to be the number 1 killer with regard to foodborne illness. It's a non-spore-forming organism, causes infections. It's found in the intestinal tract of animals. There are over 2,000 serovars. I've heard recently as high as 2,600 types. Survives freezing and dehydration. Killed by mild heat. There are some special concerns with regard to low moisture foods. It's been found to remain viable in low moisture foods. Environmental monitoring is going to be pretty important for Salmonella when you have a ready to eat food exposed to the environment prior to packaging. Salmonella, number 1 killer, number 2 cause, foodborne illness. So how can we control Salmonella? Well, Salmonella is killed easily through thorough cooking of food. One of the major problems with Salmonella is it spreads through cross-contamination. So we really need to be mindful of certain food commodities, certain products like chicken, like eggs, to avoid recontamination from the environment or surfaces. Proper hygiene always plays a factor. And low pH low pH does prevent the growth, but prevention of growth is not enough to ensure safety. So we want to make sure that we actually eliminate and kill these organisms. Salmonella. So a little bit about E. coli, non-spore forming, infectious, intestinal tract of animals and humans. Low pH prevents the growth, but it may survive, and it's killed with mild heat. So some of the things we can do to control E. coli would be good sanitation and personal hygiene, of course, preventing the spread of fecal material. It's killed by proper cooking and reheating, as we previously mentioned. Low heat can destroy or kill. Prevent cross-contamination. This is a big deal with regard to E. coli. And proper refrigeration. Low pH, low water activity can prevent the growth. Key thing to remember about E. coli is some strains of E. coli are very infectious at low doses. E. coli. So staph, Staphylococcus aureus. It's a non-spore forming organism. It does produce toxins though. It's found in the human skin or nasal passages, also on the hide and skin of animals. It's resistant to high salt. Cells are killed with mild heat. However, the toxins it produces are very heat stable. So let's look at some controls for Staphylococcus aureus. So since it's from the skin and nasal passages, it's going to be very important to prevent contamination from people. So proper hygiene is going to be important, excluding food handlers with boils, wounds, abscesses. And don't forget the vegetative cells are pretty easily destroyed with normal cooking. However, this is a toxin-forming organism. It produces toxins. The toxins are very heat stable. But we can control it with proper refrigeration, and hot holding can prevent the growth. All right, here we go. Another one, Listeria, non-spore forming, infectious, ubiquitous, we say, everywhere. Animals, humans, environment. Low pH prevents growth. Survives dehydration and freezing, though, and grows at refrigerated temperatures. Actually, studies show it grows as low as 31.3 degrees, which is actually a freezing temperature. So some controls for Listeria are going to be, again, like most, proper heat treatment, temperature control, slows the growth. Remember, it grows as low as 31.3. There is a likelihood to reduce the water activity using salt, sugar, or other growth inhibitors. Avoid recontamination from the environment, table, surfaces. And environmental monitoring is going to be big with this particular organism, especially if you're working with a ready-to-eat food that is exposed to the environment prior to packaging. So environmental monitoring will be a pretty good one to implement for ready-to-eat foods. All right, let's take a look at Campy. Campy, non-spore forming, infectious, comes from animals, primarily chicken intestines, makes its way into milk products, raw milk, animal products, water via animal contamination. So it's what we call a microaerophil, which needs a little bit of air. Grows pretty good between 32 and 45 Celsius. But this is a strange one. It dies in refrigeration. Likes pH between 4.9 and 9, but you drop the pH below 4 and it will die. So it's sensitive to heat and to drying. Campy. So one more time, Campy can be controlled with proper cooking, pasteurization, water chlorination, and of course, always looking to prevent cross-contamination, especially from raw poultry. Campy. We made our way through a bunch of bacteria. Let's take a look at some of the viruses and parasites that are infectious, like hepatitis A, cyclospora, cryptosporidium. Only some are pathogenic for humans. Most do not cross species. So primarily, they come from food handlers, fecal material, but also raw sewage could contaminate products, fruits, vegetables. They do not grow on food. I like to say they just need a ride. They just need a ride. We fail, they get on the food, food gets to the customer gets a virus or a parasite. So with regard to viruses and parasites, we really want to work on our hygiene programs, preventing contamination. But it's going to come down to source too. We want to make sure that we have specifications for our suppliers. We talked about that in the prerequisite chapter. Supplier specifications, supplier programs to ensure that the raw materials are coming in not likely to be contaminated. We can use heat treatment to kill both viruses and parasites. And parasites can be killed with freezing. So we want to make sure we use a validated freezing process for specific target parasites for that. Viruses and parasites. So we have these biological hazards: bacteria, viruses, parasites. We'll be conducting a hazard analysis on our ingredients, our food, our process. If we reveal any of these biological hazards, we'll start evaluating them, determining if they're significant. If they are significant, we'll look for ways to control them. I'll cover some of the controls again here. Things like time temperature control, freezing, cooking, refrigeration. Use of acids or preservatives. You can use HPP processing or high pressure processing. Irradiation, UV light. These are some strategies for controlling biological hazards. Let's run through a few more. We can always set standards or specifications on our suppliers. Don't forget about your food handlers, your workers, personal hygiene practices, movement. Sanitation, of course, cleaning the facility, inside, outside. We're always looking for ways to prevent cross-contamination. Don't forget about your package. How's our packaging? We want to make sure that it has good integrity, protecting the product. And we want to make sure we're looking at our storage distribution practices as a way of controlling, minimizing the growth of organisms. And one thing that we could always do is educate our consumers or provide them with directions, proper use, to help them to prevent abuse, to prevent the likelihood that they could get sick. Keep frozen until use, cooking temperatures, things of that nature. Just a few more reminders here, talking about ways of controlling pathogens. Another way we can inactivate or control them would be cooking, pasteurization, or the retort process, which is a combination of heat and pressure. And freezing is a strategy used to destroy parasites in fish. So a big part of what we do is also preventing the growth. A couple things that I want to remind you of: we are trying to eliminate, we're trying to prevent, or we're trying to reduce to an acceptable level in some cases these pathogens. So some of the strategies here would be store and prepare products under appropriate conditions, refrigerated rooms, perhaps. Prevent and minimize the growth, again, using techniques like freezing, refrigeration, hot holding. We can use salt, sugar, acid to lower the water activity to minimize growth. And then we talk about something like Clostridium perfringens. We want to make sure we're doing rapid cooling. So we've got quite a bit of this stuff figured out. I mean, we know what the enemy likes. We know what they need. We know they like food, acidity, time, temperature, oxygen, moisture, fat tongue. Some of the characteristics that enable the growth. Take a look here at some data on what temperature growths or what ranges that certain organisms like. You can see Clostridium botulinum, Listeria, Yersinia, and E. coli, and Vibrio are listed here. And these numbers are in Celsius. On the next page, we'll take a look at some numbers in Fahrenheit. So as you can see here, there's not a lot of room for failure with regard to some of these. Some actually grow in refrigerated temperatures. Clostridium botulinum, there's multiple strains or serotypes. So we got one that grows as low as 38 and one that grows 50, as low as 50. Listeria is growing as low as 31.3, Yersinia 29.7. E. coli, if we're keeping things cold the way we should, it begins growth between 44 and 46. And Vibrio vulnificus is up at 46. Keeping things cold below 40, 41. And we've got these beat for Vibrio, that is. Yeah, I rattled them off earlier. Remember, organisms like certain things. They like food. We sell food, so we're in trouble there. They like the proper acidity, pH. They like a particular temperature, time, oxygen, and moisture. Here we are looking at some time parameters that could impact the growth. At certain temperature, it needs a certain amount of time to be a problem. So here we are in Celsius again, looking at some cumulative exposure generation time parameters. One more time, talk about prevention of recontamination. So ready-to-eat foods exposed to the environment prior to packaging must be protected from recontamination. The big organism we worry about for sure is Listeria, perhaps even Salmonella. So we're going to want an effective prerequisite program to ensure pathogens, due to recontamination are unlikely to occur in the environment. Tables, utensils, and environmental monitoring is going to be the way that we can do that. Looking for the organisms after we clean, between cleaning, prevent recontamination. So I know we covered a lot of stuff here. You're probably going to need to do a little more extra studying here, a micro class, or do some specific studies on the products and processes, maybe looking at some specific organisms. I put some resources in the chapter notes, like the Bad Bug book, a little bit about prions and specified risk material. So make sure you take a look at that stuff. But this concludes chapter 3. Please complete the chapter quiz. And again, make sure you review all the attachments.